Assessing the application of pseudoviruses in the laboratory. Pseudoviruses are practical virological apparatus because of their safety and adaptability. Pseudo means fake and not infectious. Pseudoviruses mimic early stages of virus replication, including viral attachment and entry to cells. They are safe because they are an alternative viral particle for highly pathogenic viruses. They are genetically stable and essentially safer than wild-type viruses. They can be used in a biosafety laboratory level 2, while the wild-type virus may require biosafety laboratories levels 3 or 4. BSL-2 laboratory covers infectious agents that pose a health hazard. For example, we can work with pathogens like HIV and staph in a BSL-2 laboratory. In a BSL-2, there are practices that are required, such as appropriate PPE. All the procedures that can cause an infection from aerosols and splashes must be performed in a biological safety cabinet. The lab must have self-closing doors, a sink and eye wash station, and biohazard signs. Emerging pathogens like SARS-CoV-2 or Ebola required working in a BSL-3 or BSL-4 laboratories for safety reasons. Pseudoviruses can be produced naturally during an infection or artificially in the laboratory. The pseudoviral system created in the lab contains reported genome instead of the viral genome. Working with these pathogens in a BSL-2 is not allowed. Alternatively, you can produce SARS-CoV-2 or Ebola pseudoviruses displaying the viral spike on the surface of viral particles that lack the viral genome. For that reason, these pseudoviruses are not able to produce viral progeny. Instead of the viral genome, pseudoviruses contain a DNA reporter genome. Reporter genomes have genes that, when introduced into a target cell, will produce a protein or enzyme that will provide a signal for the successful attachment and entry to the pseudovirus into the host cell. Applications. Pseudoviruses can be used for conducting in vitro research on the relationship between viruses and host cell. The use of pseudovirus for in vitro research is important because it can be used to explore antiviral activity of antibodies and other antiviral compounds. They can also be used to understand better the molecular events that allows the virus to attach and enter the target cells. Pseudoviruses can also be used in vivo experiments. In vivo research involves the use of animal models to understand better the viral pathogenesis and explore the efficiency of antiviral compounds and vaccines for which pseudovirus can be produced. Pseudovirus cell-based assays are used for test neutralizing agents, neutralizing antibodies, and other compounds that trigger viral attachment or entry. Pseudoviral models using different packaging systems. The virus spike proteins such as chikungunya virus and the severe acute respiratory syndrome coronavirus use the lentiviral packaging system. The lentiviral packaging system are known for their efficiency. They are often the first preference packaging systems to develop envelope pseudoviruses. They are mainly derived from HIV-1. Another reason why it is mainly used is because they are known to retain the genetic sequence needed for viral transcription and packaging. The Ebola virus and the measles virus use the vesicular stomatitis virus packaging system. This packaging system is used because of its versatility for creating pseudotype viruses. It is ideal because it has no stringent selectivity for envelope proteins. When this packaging system is used to make pseudovirus, there may be residual VSV which is mixed with the pseudovirus, and this can produce a false positive result. Therefore, the amount of VSV used should be minimized. The influenza virus used the murine leukemia virus. This is commonly known as the retrovirus system. This is the most commonly used system to make pseudovirus. In this system, the MLV packaging codes the reporter genome and produce pseudoviruses. The production of pseudovirus in the lab. The following slide shows a method to produce SARS-CoV-2 pseudovirus. This is a method used in Professor Romero's lab. In order to produce pseudovirus, we need to transfect mammalian cells with plasmid that contains all the necessary information to produce pseudoviruses in those cells. In this case, the mammalian cell line we are using are the 293T cells. Transfection is a process by which genetic materials can be forced into a cell to express that genetic material in those cells. This figure is showing a particular transfection method that uses lipofectamine 2000. Lipofectamine 2000 is a cationic lipid that, when mixed with the plasmid, renders a complex that fuses with the 293 cell membranes and delivers the plasmid into those cells. In this figure, step 2, you can see 3 different plasmids that are incorporated into the lipofectamine plasma complex. One plasmid represents the plasmid that brings the gene that codifies for the viral spike, for the pseudovirus we are producing. The second one brings the genes that codifies for the proteins that will produce the pseudoviral capsid, and the third one is the reporter genome that contains the reporter gene that will be encapsulated into the pseudoviral capsid. The lipofectamine plasmid complex is incubated for 20 minutes at room temperature before adding the 293 T cell to the monolayer in step 3. The mixture is incubated with the cell for 6 hours at 37 degrees Celsius, 5% carbon dioxide, and 93% humidity to allow the plasmid to be delivered into the cell. After this brief incubation, the cells are washed in step 4 to remove any remaining transfection mixture, and growth medium is added to the cell to incubate them at 37 degrees Celsius, 5% CO2, and 98% humidity for 48 hours. In time in which the pseudovirus particles are assembled in the cells and delivered into the extracellular medium. The pseudovirus particles are collected from the supernatant and freeze at -80 degrees Celsius to store the pseudovirus stock until it can be used in pseudoviral cell-based assays. Pseudoviral cell-based assay. The pseudovirus display the SARS-CoV-2 spike protein on the surface, while the plasmid that encodes for luciferase is contained inside the particle, with no other nucleic acid present inside the pseudovirus. The spike protein will bind to angiotensin converting 2 receptor. The membrane of the viral particle and the cell will fuse. After fusing, the plasmid will be released into the cell, where luciferase is expressed. In the presence of the luciferase substrate, the cells will glow or luminescence. This system can be used to test the neutralizing agent that interfere with the absorption or entry of pseudovirus into the cell, and in that way block the expression of luciferase, resulting a decrease in luminescence. Limitations of pseudoviral models. Pseudoviruses contain membrane envelope proteins of the wild-type virus. The membrane envelope protein can convey the viral entry, mimicking the wild-type virus. Therefore, the pseudovirus may replicate once in some cases. This happens when the parental viral genome is modified to delete the viral genes that are required for replication and the native glycoprotein gene. And alternatively, the reporter gene coding for luciferase or fluorescence protein is inserted. When this happens, the pseudovirus are defective for undergoing a complete replication cycle and will only undergo a single infection cycle. There could be differences in the shape of pseudoviral particle versus the wild-type virus. Therefore, the glycoprotein distribution on the pseudoviral envelope may not indicate the natural state of these glycoproteins on the wild-type virus. Hence, it is wise to compare the various packaging systems to determine which one produces a pseudoviral particle that better resembles the wild-type virus. The shape of this pseudovirus can have effect on its sustainability. The use of pseudovirus proves to be an alternative approach when working with emergent viruses. They can be safely used in a biosafety laboratory too. They are a valuable tool to perform in vitro and in vivo studies. Some limitations must be considered when planning to use the pseudoviral model in our experiments.